Welcome back to Fun Facts Online. Today, we are bypassing the usual pleasantries to dive straight into a microscopic mystery that has baffled scientists for over 140 years. We are looking inside the cells of the malaria parasite to witness a frenetic, physics-defying dance. Within the digestive belly of Plasmodium falciparum, the deadliest of the malaria parasites, there lie tiny, jagged crystals of iron. Under a microscope, these crystals do not merely drift. They spin, they tumble. They ricochet with a violence that defies the calm viscosity of the cell's internal fluid. They look like microscopic rotors, whirling engines of iron locked in a perpetual struggle. For decades, this phenomenon was a biological curiosity, a blind spot in parasitology known as the dance of the hemozoin. But recently, in a groundbreaking shift that bridges biology, physics, and nanotechnology, we finally understand why. These are not just passive crystals jostled by thermal energy, they are active machines. They are Ferrous rotors, powered by a chemical reaction that mimics the propulsion of rockets, serving as the first known example in nature of a self-propelled metallic nanomotor. To understand this discovery, we have to start with the deadly architect itself. Malaria remains one of the most devastating infectious diseases on Earth, and the culprit is a single-celled protozoan parasite of the genus Plasmodium. When an infected Anopheles mosquito bites a human, it injects sporozoites that travel to the liver and eventually the bloodstream. This is where the clinical nightmare begins. The parasite invades red blood cells, hiding from the immune system within the very cells designed to carry oxygen. Once inside, the parasite is a voracious eater. It consumes up to 80% of the host cell's hemoglobin. However, this feast comes with a heavy price. Hemoglobin digestion leaves behind a toxic scrap called heme. Heme is the iron-containing component that binds oxygen. In its free state, unattached to protein, heme is a molecular grenade. It is highly reactive and capable of generating free radicals that shred cell membranes. For the parasite, digesting hemoglobin is like eating a cherry bomb, nutritious on the outside, but explosive at the core. To survive this toxic influx, the parasite stitches these free heme molecules together into chemically inert crystals called hemozoin. These dark brown crystals accumulate in the parasite's digestive vacuole like a growing pile of trash. For over a century, scientists viewed hemozoin as just that, a static dump of iron. But they were wrong. The trash pile is alive with motion. The observation of this pigment actually predates the discovery of the parasite itself. In 1847, German anatomist Johann Heinrich Meckel observed black-brown granules in the blood of patients. But the true breakthrough came in 1880 in a military hospital in Algeria. A French army surgeon named Charles-Louis Alphonse Laveran was examining fresh blood smears. He saw the pigmented bodies inside the red blood cells, but he also saw them moving. He described filaments whipping around, and the pigment granules dancing with a frenzied energy. This animation convinced him that he was looking at a living organism, not a bacterium. The dancing pigment was the first clue that malaria was a parasitic disease. For the next 140 years, this motion became a diagnostic marker. If you saw the jiggling dark pigment, you knew the parasite was alive. Yet, despite its utility, the mechanism was dismissed. Most physicists and biologists assumed it was Brownian motion, the random jittering of particles suspended in a fluid caused by collisions with water molecules. It was an easy assumption to make. In the microscopic world, everything jiggles. But careful observers noted something odd. The hemozoin crystals moved too fast. They tumbled and spun with a vigor that seemed to exceed the passive thermal energy of their environment. They looked less like dust motes floating in sunbeams, and more like angry hornets trapped in a jar. It wasn't until late 2025 that a team of researchers led by Dr. Paul Segala at the University of Utah published a study that shattered the Brownian motion hypothesis. Using advanced high-speed imaging, they tracked the movement of individual crystals inside live parasites. The data was undeniable. The crystals were moving significantly faster than the laws of physics allow for simple diffusion. There was an active force at play. But what could push a rock inside a stomach? There are no muscles in the digestive vacuole. The answer lay in the chemistry of the crystal surface itself. The researchers discovered that the hemozoin crystals are catalytic surfaces. The digestion of hemoglobin releases not just heme but also reactive oxygen species, specifically hydrogen peroxide. In most cells, enzymes like catalase quickly break down hydrogen peroxide because it is toxic. But the malaria parasite lacks catalase in its digestive vacuole. Instead, it relies on the hemozoin crystals. The iron on the surface of the crystal acts as a catalyst. It facilitates the breakdown of hydrogen peroxide into water, oxygen gas, and energy. This is an exothermic reaction. In the macroscopic world, the rapid decomposition of hydrogen peroxide is used as rocket fuel. In the microscopic world of the parasite, This same reaction creates tiny, localized gradients of oxygen and fluid flow. As oxygen bubbles form or fluid is ejected from the crystal surface, it generates a recoil force. Because the crystals are irregular in shape, jagged, brick-like or rod-like, the force is not uniform. It strikes one side harder than the other, creating torque. The result is that the crystal spins. It acts like a tiny, unguided rocket, careening around the digestive vacuole. To appreciate the elegance of this, we must look at the physics of the very small, a realm governed by low Reynolds number hydrodynamics. For a human swimming in a pool, inertia matters. If you stop kicking, you glide. But for a microscopic object like a hemozoin crystal, inertia is negligible. In this world, water feels as thick as molasses or tar. If a microscopic swimmer stops its engine, it doesn't glide. It stops instantly. To move, you must constantly apply force. The hemozoin crystals are overcoming this immense viscous drag through continuous chemical explosions. Physicists classify this as active matter. Unlike passive matter, like a dead leaf floating in a stream, active matter consumes energy to generate motion. The hemozoin crystals are a rare example of non-living biological active matter. They are inanimate objects that behave like living things because of their chemical environment. But why do they spin? Evolution rarely maintains energy-intensive processes without a reason. The researchers propose two critical survival functions for this Ferrous rotor mechanism. First is the blender effect. Crystallization requires surface area. The parasite needs to add new heme molecules to the growing crystals constantly. If the crystals were static, they might settle and clump together, burying their active surfaces in a pile of sediment. By spinning wildly, the crystals keep the digestive vacuole agitated. They act like the impeller in a blender, preventing clumping and ensuring that every face of the crystal is available to bind more toxic heme. The second function is the afterburner effect. Hydrogen peroxide is toxic to the parasite, just as heme is. By using the crystals to break down the peroxide, the parasite solves two problems at once. It uses the dangerous peroxide to power the motion needed to dispose of the dangerous heme. It is a masterstroke of metabolic efficiency. Beyond their motion, these crystals possess unique physical properties. They are paramagnetic and dichroic. Most biological materials are diamagnetic, meaning they are repelled by magnets. However, hemozoin is paramagnetic. The iron atoms in the crystal lattice have unpaired electrons that align with an external magnetic field. This means if you place infected blood in a strong magnetic field, the random tumbling stops and they snap to attention. They are also dichroic, meaning they absorb light differently depending on their orientation. This creates a blinking effect that can be detected with lasers, forming the basis of magneto-optical diagnosis, a method that can detect a single parasite in a drop of blood in seconds. This discovery opens up entirely new avenues for treating malaria. If the spinning motion is essential for survival, then stopping the spin should kill the parasite. We could design drugs that act as catalase mimics, breaking down the peroxide fuel before it reaches the crystal. Or, we could use surface blockers to gum up the catalytic sites. Furthermore, this study of natural nanomotors is inspiring engineers. If nature can build a self-propelled, iron-based robot that runs on blood byproducts, perhaps we can build synthetic ferrous rotors to hunt the parasite. These artificial magnetic nanoparticles could be guided by external fields to deliver drugs directly to the source. In conclusion, the malaria parasite is not just a biological entity, it is a microscopic engineer. It builds crystalline machines, fuels them with oxidative waste, and uses them to churn its own stomach. The spinning iron crystals of malaria teach us that even in the chaotic interior of a cell, physics rules. And by understanding the rules of that physics, the hydrodynamics of the spin, the chemistry of the fuel, and the magnetism of the crystal, we may finally find the wrench needed to break the machine and end the scourge of malaria once and for all. The dance of the hemozoin is no longer a mystery, it is a target, and the music is about to stop.